Hello, guys. How are you today?
Doing well.
Pretty well. So I guess you can hear me, which is good. I'm gonna finish chapter 11 today, and you will have the opportunity to earn some extra points, right? By taking the extra credit quiz. So you have the information about it. I sent it out last night. So as I
What?
mentioned,
I'm so sorry to interrupt. What will that? What material will that cover or go over?
yeah, basically all the questions are pretty simple. So it will. I don't think it will require any special preparation. So if you did reasonably well on exams 1 and 2, but it will also cover material that we've discussed in lectures up to today, up to this week, basically.
Perfect.
So
Thank you.
it will not have chapter 12, but it will have all the other chapters. Yeah, so if you did well on the test, so I don't know if it requires any special preparation. If you didn't do well on the exams, then you may not do so well on this one either. So this is some information for you to digest. So you may want to review material a little better in that case, just to be able to earn those points. And you have time. You have time. Today is what, Wednesday. You can you have from Wednesday to until Monday morning. So yeah. And those of you who have accommodations, you have some extra time to do it. And all right. Anything else? I will also have a review, final review meeting with you guys. I'll send you the date for that. I haven't started making the final yet. So once I have it pretty much put together, then I'll be ready to do that review for you so that you know that I'm prepared for it, not only you. And so I'll send out the date. If you can't attend that, it will be recorded, of course. So, but you will not be able to ask questions if it's recorded. But you can always participate in a discussion forum. And one thing about the discussion forum, there's not so much activity there, but I encourage you to use it more. It's a great way to learn the material. So, all right. So let's go to chapter 11. Share screen. All right, so we're talking about lipid bilayer membranes. And we stopped around this part on, I think, slide 33. And we talked about different lipid composition of membranes. And we know that the melting temperature of the membrane determines its fluidity, right? And melting temperature will decrease as double bonds are added. We talked about that, and melting temperature will increase as you increase the length of the saturated fatty acids, right? So you increase the intermolecular interactions, van der Waals interactions, and melting temperature will increase. And so the interesting thing is that the cells can regulate their membrane fluidity by adjusting their saturated fatty acid makeup, right? So at high temperatures, they will need long saturated fatty acids, right? So that this cells don't freeze, cell membranes don't freeze. No, so the cells do not become too fluid and resist those high temperatures. At low temperatures, cells need more unsaturated fatty acids, which will not freeze. So makes sense, quite logical. All right, so also membrane rigidity and permeability can be regulated by steroids. We talked about cholesterol, which fills the gaps in cell membrane in eukaryotic cells. Ergosterol, this is a similar molecule which performs a similar function in fungi. So we talked about yeast, for example, and in plants. This is actually in prokaryotes, aerobic prokaryotes who have hopanols. Again, there's a multi-ring system, similar to cholesterol, but has 5 rings and some kind of polyhydroxy tail. Okay, so and when we talk about fluidity, what does it mean? Well, it turns out that these lipids can move in as a lateral diffusion, right? They can move very fast, 1 micrometer per second. And in fact, this is close to circumference of one particular cell, a bacterial cell, for example. For example, you can see that a lipid can circumnavigate an E. coli cell in 1 second, right? So in other words, the lipid can leave, go around, come back in 1 second. It's amazing how fast these lipids move one past another. So when we study them, when we study embedded proteins, the lipid composition, we kind of have this, we treat them as like some rigid structures, but they're not rigid at all. They're like a sea, like an ocean, right? With species, with lipids moving one past another very quickly. Okay, so how do we know that? So there are many techniques. One of them is known as a photo bleaching, known as fluorescence recovery after photo bleaching, FRAP. FRAP, and what does that do? So this is the general picture, but let's look at the specific steps. So, so this is again step 1. So this is our cell, lipid bilayer. We're going to apply a fluorescent probe, some kind of dye. Okay, these little red dots will not penetrate into the bilayer, but they will form complexes with the head groups. Okay, and then we're going to irradiate that with laser and bleach it. So in other words, so here's the view of the fluorescent microscope. You can see there's all these cells there's no cavities, there's no empty spaces. But then we're going to irradiate with the laser and we're going to bleach it and create this kind of empty area where no dye is present. No dye is present, and then allow it some time so that the phospholipids will migrate and actually repopulate this empty space, and the dye will migrate together with the phospholipids because it's attached to the head groups, right? So we're gonna measure the rate of fluorescence return. And by the rate of the fluorescence return, we know how fast these things migrate in the lipid bilayer. So, you have any questions? So this technique is known as FRAP. It's very easy to understand. And that's how we can have an appreciation how fast these lipids move in lateral direction. Now, it's also possible for the lipids to flip from the outer leaflet to the inner leaflet and the other way around. So this is known as a flip-flop diffusion, right? And you can see that it's actually much slower now. So half-life of a lipid in one leaflet is measured in days, right? So no longer seconds, it's days. Why do you think that is? Why do you think it takes so much longer for the lipids to flip from one leaflet to the other compared with the lateral diffusion? Why does it take days as opposed to seconds? Just think about it. Think about it. Think of, based on what you know about the structure of lipids.
Would it be because of its hydrophobic, like its interactions,
Interactions, yes.
like it's the molecular interactions, like van der Waals? Is it van der Waals?
So what species interacting with each other? What are you what do you have in mind? You mean this is stuck because of these interactions with other lipids?
I would say so. In
But-
order for it to break apart.
But that would be the case when we have lateral diffusion, right? When they move in this direction.
Mm-hmm.
But so why would it play a bigger role in the flip-flop action?
Could it be their interaction with like transmembrane proteins?
Transmembrane proteins, maybe, but let's say there are no proteins. It'll still take days. Just think about, yeah, go ahead.
I was just going to say, what about the interaction that it's having with like what's outside of the cell or inside of the cell? Because it would have to go to the other side. So those interactions would be stronger, right? Because it's the polar head. So it's those interactions like with the outside or inside of the cell.
Maybe it's length as well.
No, it's not linked. But just think about it. So you have a polar head group. For this to undergo this kind of flip-flop, what needs to happen? This polar group has to come inside of the bilayer. That's the only way, right? You have to drag this lipid inside for it to undergo this kind of transition, which means the head group has to come together for the ride, right? And so obviously this head group, which is charged, does not want to be inside the membrane. So it's the fact that the head group is stuck on the outside and doesn't want to go inside. I think that's what prevents it from this kind of transverse or transition from the outside to inside and vice versa. So for this kind of diffusion, there are enzymes that catalyze this kind of diffusion. And just as the general name for these enzymes are flipases, but there are actually different kinds of flipases. And some of them use the energy of ATP, right? ATP, adenosine triphosphate, which undergoes hydrolysis. So you can see here, this flipase known as a P-type ATPase. ATPase usually means an enzyme that uses the energy of ATP to perform a function. And so this one will move phosphatidyl ethanolamine and phosphatidyl serine from the outer leaflet to the inner leaflet, right? Then there's a flopase, which actually does exactly the opposite. Usually these are ABC transporters. So we'll talk about ABC transporters a little later, but they also utilize the energy of ATP. So here the lipid will be moving from the inner to the outer. And there are scramblases. Scramblases are those that don't use any energy. They just basically allow for the lipids to move from one leaflet to the other based on their concentration gradient. So in other words, if there is a lot of certain type of lipid on the inner side, the scramblases will allow the path, will give them a path to undergo this kind of flip-flop movement toward evening out of the concentration gradient. Okay, makes sense. Now, obviously, now I already mentioned that. Let's see if you remember phosphatidylserine. Where does that lipid live? In the inner membrane or in the outer leaflet?
Isn't it on the inner and then it moves to the outer if the cell needs to undergo apoptosis?
All right, that's right. And also I think in platelet activity, also important. So phosphatidylserine is a dangerous lipid. It has to be kept on the inside. If some of it gets out and into the outside, then this cell can be eaten by phagocytes and goodbye, right? So cells have to consume energy to make sure to keep all their phosphatidylserine inside. Who would know, right? Just a particular lipid can be a death signal. All right, another interesting feature about cell membranes is the formation of what's known as rafts. Rafts, just like literally rafts, these are, if you imagine that the rest of the membrane is an ocean, so these will be rafts which float in this ocean. And the rafts are basically they're rich in sphingolipids and cholesterol. And you can see these sphingolipids, they will have longer fatty acid chains, right? And because of the longer fatty acid composition, they kind of, the thickness of the bilayer is a little larger in the raft, right? And you can see the red ones, red spheres are sphingolipids here. And so they're also rich in some special proteins, for example, GPI-linked proteins. So we talked about that. And so these rafts, depending on the cell type, there actually could be few, it could be lots. There are certain cells where rafts can constitute up to 50% of all the area on the surface of the cell. So it's just basically half raft, half ocean, right? And lots of cholesterol as well. So these rafts are quite rigid. And they actually, because of their rigidity, they can actually provide home for many proteins. And so there are specific proteins which live specifically in rafts, and they interact with one another. They interact with one another because they're brought in close proximity by rafts. Okay, so actually this is a great area of research right now, as it is believed that rafts play a huge role in many physiological processes. All right, let's look at membrane fusion. So, which is a process in which the two membranes can fuse without spilling the content, right? So here we have what's Golgi complex. Remember, Golgi complex is a site where proteins are covalently modified, for example. And you can see there will be a vesicle that will bud off the Golgi, fuse with the cell membrane, and allow for some kind of secretion process, like some kind of modified protein through exocytosis will leave the cell and travel to a distant part of the body. Now, exocytosis is the departure of something, right? Endocytosis is when things come into the cell. Now, endosome and lysosome, right? So here, for example, things which arrive, things that need to be degraded, right? So there's a membrane kind of invagination like this, and the species will accumulate inside of this invaginated area, and this membrane will fuse together with the lysosome membrane, and the content will come in to the lysosome, and lysosome, you know, at low pH and high concentration of proteolytic enzymes. So these proteins will be degraded. Viral infection, right? So virus, its membrane will also fuse with the host cell, sperm and egg, vacuoles in plants, and also separation of two plasma membranes at cell division, right? So So there are many different processes how this happens, and we're going to look at one of them, which involves snare proteins. Snare proteins, so specifically it's involved in neurotransmitter release. So there are actually 3 types of snare proteins: T, V, and Q. And what do they do? So let's look at this bigger representation. So let's say this is the secretory vesicle with the neurotransmitter, right? So just think about it. How does neurotransmission occurs? So you have presynaptic neuron with vesicles with neurotransmitter, right? And there's a membrane, obviously, like so. And there is a postsynaptic neuron like so. So these vesicles, right, which also have membrane, they have to fuse with the cell membrane without spilling the neurotransmitter, right? For the neurotransmitter to come out and transmit the nerve signal to the postsynaptic neuron, right? So that you can get the action potential started in the postsynaptic neuron. So these snare proteins, so V snare stands for the vesicle, T snare stands for the target, right? Target. And there's also SNAP25, which is the third major player in this process. And so what you can see here is that these 3 are starting to form a triple helix. The triple helix will catch both and bring them in close proximity, right? So this is the cell membrane of the neuron. This is the vesicle membrane. And so eventually they will bring them into close proximity and the 2 membranes will start interacting. The lipids will, and this triple helix will start tightening, tighter, and tighter. And the 2 membranes will start, the lipids will start to undergo exchange, and it will create an opening here. It will create an opening. It will create an opening until basically the 2 membranes will fuse, right? And there'll be one free space here. So this is known as hemifusion. Hemifusion, where the outer leaflets of both membranes come into contact. But the neurotransmitter still has not left. It's still sitting there. And so this will need to proceed further to complete fusion, where now the 2 membranes will, the inner membranes will also fuse and allow for the neurotransmitter to spill out. Right, so highly organized process. And so the membrane fusion is very important for the physiology of physiological processes in the body. All right. Okay, transport across membranes. So this is going to be the major topic for the rest of this lecture. So we're going to have, so normally cell membranes are permeable, small non-polar molecules, right? But the polar molecules, unfortunately, do not cross the membrane so well, right? Makes sense because the membranes have fatty acids. They're non-polar. So polar molecules cannot cross the cell membrane easily. And so transport of this membrane, but the transport can be facilitated by proteins that provide an alternative diffusion path. Such proteins are known as transporters. And so transport must be energetically favorable, otherwise obviously the molecules will not cross the membrane. There will be concentration dependence, right? So the solute will move toward the equilibrium across the cell membrane. So you can see here, C1, the concentration on the left hand side is larger than C2. And if you have the diffusion path here, there's a protein that allows you to get across the cell membrane, the molecules will start crossing toward down the concentration gradient. And eventually the process will continue until equilibrium is achieved. Now this is electrochemical dependence. This is toward the charge, right? The species don't have to be the same. They can be different as long as the charges are balanced. So you can see there are more negative charges on the left, right? And there's more positive charges on the right. And so you can see here that the negative charges will move in this direction, the positive charges will move in that direction. So there is electrochemical potential until it becomes zero. At equilibrium, it will become zero. So the sum of the, so the charges have equalized. So there's no net charge on the left, no net charge on the right. Okay. So polar solutes, they need alternative paths to cross membranes. So you can see here, what's the major difficulty here? The major difficulty with polar solutes is that they're hydrated. They're hydrated in water. Remember, likes like, right? So if this is some kind of alcohol, for example, it will be highly hydrated. And so what needs to happen once it starts crossing the membrane, it cannot bring all these molecules of water with it, because water and lipids do not mix. So this molecule has to kind of shake off the hydration sphere, become kind of naked. And then in a naked state, it can cross the membrane. Once it leaves on the other side, it immediately regains the hydration sphere, hydration shell. And so this process of shaking off the molecules of water is accompanied by very high activation energy, right? So here in red, you can see here without the transporter, we have to go all the way up in energy like that, cross the membrane, and then we can actually drop once the hydration sphere is regained. Now, if you have a transporter like this, what can happen is that, yes, this will still drop all the molecules of water, but it will be compensated. So this transporter will have a lot of polar residues inside the channel like this. And so these polar residues will interact with the polar residues of your compound of interest. It's kind of water bonds, hydrogen bonds, will be replaced by bonds within the transporter. And so once it reaches the outside, then it will regain the hydration sphere and it can leave. And so you can see here, the path is in blue, and there's very low activation energy associated with that. All right, so types of transport. Types of transport. So first we have a simple diffusion. So this will be for nonpolar compounds only, down the concentration gradient. So the lipid bilayer is not a barrier for these kind of compounds. Now, if the compound is polar, but still there is concentration gradient, right? Electrochemical gradient. You can present it with the protein that will allow for the passage. And again, from high concentration to low concentrations, the molecule can pass through. If the concentration gradient is unfavorable, so let's say concentration on the inside is higher than on the outside, how do we bring the molecules inside? So this will be primary active transport against the electrochemical gradient. And so here we're going to use ATP to do that. we're going to hydrolyze ATP, and that will allow for the molecules to come in. And there is a secondary active transport. So for example, it can also use the energy of ATP, but it can also use a different molecules. For example, if there is ion gradient, right? So let's say if there is high concentration of ion on the outside, right? And low concentration of ion on the inside. And so the ion will be coming in, and the solute can use that if that transport is coupled with the movement of ion, the solute can move from the outside to the inside. Then we have just simple ion channels. So these will just open up and the ion will move down the concentration gradient. Now this can be gated. Gated means the ion channel can be closed, and it can be closed in the presence of a particular ligand or in the presence of a different ion, right? So this ion can open and close, allowing this ion to come in or not. And there's also ionophore mediated ion transport down the electrochemical gradient. So this is the mechanism employed by various microorganisms, for example, right? So for example, many bacteria need iron for their survival. How do they get iron? They need to get iron from the extracellular media, right? And they synthesize molecules known as ionophores. Ionophores, they synthesize those ionophores go out, go hunting basically, right? So they're empty, they empty, and then they encounter some kind of iron outside. Iron 3+ usually, ferric ion, which will go inside. And now you have a loaded ionophore. So this will, once it's loaded like this, it can cross the cell membrane, bring the ion inside, release the ion, and then go back outside hunting for more iron ions. So this is ionophore mediated ion transport. So highly prevalent in the bacterial world. And also there's a lot of activity now in drug discovery, discovery new antibiotics that will actually inhibit this process, right? Inhibit this process, and thus the bacteria will die because the bacteria do not get enough iron, for example, right? So these ionophores need to be synthesized by bacteria, and if you inhibit the synthesis of ionophores with a drug, then the ionophore will not help the bacteria acquire iron, and the bacteria will die. All right, any questions? So this is general view. So a little more about some general things. Now, if the solute comes in by itself, nothing else is involved, it's called uniport. Now, if this solute comes in with something else together, it's co-transport. Co-transport, it's known as a symport. And co-transport can also occur if the solutes go in opposite directions, that's antiport. And again, that's very useful if, for example, one of these substrates goes down the concentration gradient, and then the other can be brought in even against this concentration gradient. And we will see some examples. So glucose. Glucose needs to, lots of cells need glucose from the bloodstream, right? So for example, erythrocytes heavily depend on, remember erythrocytes are red blood cells, right? Heavily depend on glucose as nutrient. Now blood-brain barrier, so brain, and specifically the glucose has to cross blood-brain barrier to get into the brain, right? And so, and glucose by itself is polar. So how many hydroxyl groups does glucose have? Can you count? Obviously, hydroxyl groups will make glucose very polar, right? So how many hydroxyls? Let's see if you can close your eyes and picture the molecule of glucose in your mind. And you get how many?
Is it 5?
5. Good job. So you have 5 hydroxyl groups in glucose. Obviously, these will not allow glucose to cross cell membrane. And for that reason, there are transporters, which are proteins referred to as GLUTs. Stands for glucose transporter, very easily. And so these two, erythrocytes and brain, which is surrounded by a thick membrane known as a blood-brain barrier. So those utilize transporters number 1, GLUT1. GLUT1. Now, for example, there's also GLUT2. GLUT2, which occurs, for example, in the epithelial cells of the in intestinal epithelial cells. Intestinal epithelial cells. All right. So they differ by amino acid makeup, but their function is the same, is to bring glucose. And so the way they work is, so for example, this one, I think it's GLUT1, it has 12 transmembrane domains. You can see here the yellow, so basically these are alpha helices. The yellow ones are hydrophobic amino acids, which are necessary for this GLUT to be embedded in cell membrane, right? If you don't have hydrophobic amino acids, there is no reason for the GLUT to stay in the cell membrane. But it also needs polar residues. So most of them are blue. And there are even some charged ones, but the charged ones are primarily on the outside. Obviously, they don't like to be inside. And so what happens, these blue ones actually come from, so this is a view of the alpha helix in the GLUT transporter from the amino terminus end. And you can see here, so this helix is what's known as amphipathic. Amphipathic. So basically what that means is that one side of the helix will be composed of the polar residues, and the other side of the helix will be non-polar. Remember, one helical turn is 3.6 amino acids, right? And so if you every third and fourth amino acid is polar, then and the others are non-polar, then you can create this amphipathic helix. And why is it necessary? Why is it necessary? When these helices come together like this, right? You can see they can organize themselves in such a way that inside you will have the polar sides of the helix, and on the outside you will have non-polar sides of the helix. So non-polar sides of the helix will interact with the lipid bilayer, which will anchor the helix in the membrane, and on the inside you will have polar, which will have an opening inside and allow glucose to pass through. Now this is just a image where from the X-ray structure of one of the GLUTs, which was cut in half. So you can see that there is an opening inside that allows for glucose to come in. And the way it works is very simple. So it has a number of states. You can see here, state T1, where there's the glucose binding site is open. The glucose comes in and occupies this binding site. So that's the step one. Once the glucose is trapped, the intracellular side opens up, and that's step two. The glucose departs, step three, and then again transformation, again the conformational change, and it goes back to state T1, ready for the next molecule of glucose. All right, do you have any questions about glucose? All right, we'll actually see glucose transporter one more time before the end of this lecture. Now, bicarbonate transporter. So this one is an antiporter. And so the goal of this is to provide carbon dioxide with a path to leave the body, leave the tissues, go to the lungs, and to be exhaled in the lungs. And so carbon dioxide by itself is not well soluble, in water. As you remember, carbon dioxide has no dipole moment, right? Has no dipole moment. So the two internal bond dipoles, they cancel each other out. And so carbon dioxide not very well soluble in water. And so what happens is their erythrocytes what they do, they have an enzyme known as carbonic anhydrase. So what this enzyme does, and basically it grabs carbon dioxide, which is produced by catabolism. By the way, what's catabolism? Anybody knows catabolism? So there's a common name, metabolism, when species undergo, especially small molecules undergo transformation inside the organism. But then metabolism is divided into two, catabolism and anabolism. What's the difference?
Isn't catabolism breaking down into smaller molecules, yes, whereas anabolism is like building up into bigger molecules?
Yes, exactly. And so catabolism, which will be produced, which will produce carbon dioxide, right? Basically burning of the fuel will produce carbon dioxide. Carbon dioxide will react with water in a reaction catalyzed by carbonic anhydrase. And remember the immediate product of this reaction. So if you react carbon dioxide with water, the immediate product is carbonic acid, which then undergoes dissociation. Dissociation. And so you can see here, now we have bicarbonate, right? And so now carbon dioxide is transformed into bicarbonate, which can leave into the bloodstream. Now it's fully soluble. But the problem is, that if bicarbonate just simply leaves, that would create electrochemical potential. So in other words, it will not leave so easily because we unbalancing the charges, right? Because this is a charged species. And what happens, there is a chloride that comes in and takes place of the bicarbonate in the erythrocyte. And the negative charge leaves and another negative charge comes in. And the sum of the charges do not change on the outside or inside. Then once this in the area near the lungs, in the blood vessels that are found in lungs, this bicarbonate comes into the erythrocyte and the chloride leaves. The chloride leaves, it's released, and the enzyme will do the opposite. Remember, the enzymes catalyze the forward reaction and catalyze the reverse reaction at the same rate, right? So here it grabs bicarbonate proton, regenerates carbon dioxide, which leaves the erythrocyte and then is exhaled through the airway. So, so this is an antiport system. Okay. All right, this is a really neat one. This one is Symporter. This one is Symporter, which is also designed to provide a path for the glucose to cross epithelial cells, which line the gastrointestinal tract, specifically the intestinal portion of it, right? So you can see here, so there's a glucose that comes with the food, and here's the glucose which ends up in the blood, right? So this is intestinal lumen, right? And apical surface, basically on the top of the microvilli. What are the, so these microvilli are these kind of invaginations, right? What are microvilli for? Do you know?
The absorption of broken down nutrients.
So why do we need microvilli? Well, we can absorb nutrients without microvilli, right?
It's for increasing surface area.
Yeah, makes sense. So microvilli basically allow for larger surface area for this kind of stuff, for the glucose to come inside the epithelial cells of the intestines. All right. So so how will the glucose come in? So here it will utilize a Symporter system, right? 2 sodium. So it turns out that sodium on the outside, its concentration is higher than the concentration of sodium inside the epithelial cells, right? And why is that? Can you look at this picture and tell me why how the concentration of sodium is maintained low inside the epithelial cell?
Due to the sodium potassium pump?
That's right. Sodium, potassium, and ATPase, which is uses the energy of ATP, right, to bring 2 sodiums into the cell, and 3 sodiums into the cell and 2 potassiums outside the cell. So in general, this is actually, so it's very common in all cell types to maintain the concentration of sodium high on the outside and low on the inside. In general, in most cell types, if you have a, let's say, a cell, right? If you have a cell, so inside the cell, the concentration of sodium is 12 millimolar. and the concentration of potassium is 140. Millimolar. On the outside, it's reversed. The concentration of potassium now is small, 4 millimolar. The concentration of sodium (145). Millimolar. All right. And so that's due to the action of sodium potassium ATPase. So it's actually it's one of the most energy consuming enzymes in the body. So, that keeps the concentration of sodium low. And you can see here in the glucose, once the concentration builds up in the epithelial cell, it just simply goes out down its concentration gradient using the GLUT2, right? So it's a glucose uniporter, GLUT2. Okay, any questions? All right. Now for active transport, now there is for primary active transport, so we just saw the sodium potassium ATPase, that's active transport, right? So in general, for there's a primary active transport, and here you can see that energy of ATP will be used to transport the solute against its concentration gradient, right? So here the concentration of this is small. Concentration of this is large. And so you can still pump things out because you're consuming the energy of ATP. But there's also secondary active transport where you can see here the solute, so S2 is, let's say, what we want, right? So its concentration is small. No, sorry, its concentration is large. Concentration is large. And here its concentration is small. And so this system will utilize ATP, and so it's going to be relying upon another molecule, S1 or ion. So this will be pumped out of the cell, come in, and then will be co-transported in a symporter mechanism inside the cell together with S2 and bring S2 into the cell. And S1 will just continue circulating. So in a way that we just looked at this symporter, this is similar, right? So the glucose is imported into the cell due to the low concentration of sodium that is pumped out of the cell by sodium potassium ATPase. So this is, in a way, it's a secondary active transport mechanism. All right, ABC transporters. So these are highly prevalent in many different cell types. And their role is actually to pump out or bring in, depending on what function they serve, again, small molecules. If they pump something out, that will be toxins, drugs, for example, toxins, products of metabolism, right? And so some of them are specific. and others are more what's known as promiscuous, where they basically non-specifically pump out any kind of small molecules of the cell non-discriminately. So they have a homologous transmembrane, two parts of transmembrane domains. These are, let's see, 1, 2, 3, 4, 5. So I guess each composed of 6 helices, I think. So these will be transmembrane alpha helices. And there is an internal cavity, which allows for the molecules to pass through. And there are 2 nucleotide binding domains. So this is for ATP, where ATP will bind, right? Now, this is a homologous one found in bacteria. So there's also nucleotide binding domains and transmembrane domains. So they homologous, they function very similarly. And let me go to the next slide, but then we'll come back to this one. So, so the way they work is, so let's say there's a substrate that comes in, right? It's open on one side. Now nucleotide binding domain are brought together. They're brought together by ATPs. Now ATP, once this substrate comes in, this will undergo conformational change. ATP will be hydrolyzed into ADP, but ADPs are now will not hold the transmembrane domain, sorry, nucleotide binding domains together, which will result in the opening, and the substrate can pass straight through, right? And then the process will again continue on. The ATP will come in and replace ADP. We'll replace ADP. And the system will go back and ready for the next molecular substrate. Now, so these ABC transporters, they are actually very important in cancer cell resistance to drugs. So a phenomenon known as multi-drug resistance, MDR. Multi-drug resistance. So, so cancer cells, they overexpress these ABC transporters on their cell surface to pump out drugs, toxic drugs that can destroy them. So oftentimes what happens, let's say a patient is diagnosed with a particular type of cancer. The tumor is localized, so it can be treated with surgery, radiation, and then chemotherapy. Let's say first chemotherapy will be done with a drug, doxorubicin. So it's a general cancer therapeutic agent. And it works, the tumor is gone, right? So the patient goes home, the tumor is gone. Then 2 years later, unfortunately, the tumor comes back. The patient goes for the second treatment. And then all of a sudden, the tumor does not respond anymore to the treatment with doxorubicin. So the doctors say, okay, so the cancer cells may have developed resistance to doxorubicin, but let's try something else. Let's try a different drug then, which has a different mechanism, different mode of action, different structure, paclitaxel. And but all of a sudden, the cancer cells do not respond to that either. Then try a different one, and it's again the same result. So that's what multi-drug resistance is about, and it comes from the fact that these ABC transporters are so non-specific. So it doesn't matter to them if it's doxorubicin, if it's paclitaxel. They will pump it out of the cell and will not allow for them to perform its cytotoxic action. Now, there's been a lot of research efforts on trying to design inhibitors of these ABC pumps. Now, oftentimes in the literature, they refer to as P-glycoprotein. P-glycoprotein. And unfortunately, everything that has been evaluated clinically has been too toxic to normal cells. And it's probably because ABC pumps, they have so many functions, physiological functions, that inhibiting them simply is not a viable strategy to treat cancer. Questions? Nope. All right. So this one is also a type of ABC transporter, not exactly, but similar in a way. But this is more of an active transport of an ion. Active transport of an ion, specifically chloride. So this CRFT transporter, so it's a chloride, some kind of chloride regulator. Yeah, I don't remember the abbreviation, but it's not the point. The point is that, so this is an active transport of chloride. And so the way this works, there's this regulatory phosphate, which is on serine hydroxyl, right? Once the phosphate is on the serine, it allows for the two molecules of ATP to come in and bind to the nucleotide binding domains, NBD1 and NBD2. Once the ATP is bound, this allows for the pumping of chloride outside. So the channel is open. And once that happens, then the channel closes up due to the hydrolysis of ATP and departure of the phosphate. All right. So ATP is hydrolyzed, phosphate departs, and the channel is closed. Now the cycle again restarts when the serine is phosphorylated again. Phosphorylation leads to the ATP, and another chloride is pumped out after ATP is hydrolyzed, and the channel closes. So this protein is found in many tissues, but it's widely spread in the lung tissue, where basically it allows for the chloride to enter the lungs, basically. And as the chloride does so, it, due to osmotic pressure, it brings water with it, right? And so the lungs become hydrated, right? So there's more water in the lungs, and that allows for the mucus clearance. So when you cough things up, right, that water will allow for the mucus to rise through the airway, and you can get rid of it. If the chloride concentration stays low in the lungs, then no water will come in either. No water will come in either, and the mucus will get stuck in the lungs. The mucus will get stuck in the lungs, and that will give rise to, that will provide a feeding ground for a lot of different types of bacteria. One of the most pathogenic one is Pseudomonas aeruginosa. And so it turns out that, so there are children that have defective transporter right? So there's amino acid mutation, one particular amino acid mutation that actually doesn't really interfere with the function of this channel, but makes it susceptible to degradation. So in other words, this protein is transported by itself into these lysosomes where it's degraded by proteases, because the cell thinks that this is a defective protein and we need to get rid of it. And so its presence is diminished in the lungs. And so these children, they're afflicted with the disease referred to as cystic fibrosis. Cystic fibrosis. So it's a genetic disease. A lot of people actually carry this mutation in one of the alleles, but those that carry in both, they usually do not survive long enough into the adulthood. So usually, that's why we refer to these patients as children with cystic fibrosis, because most of them will not become adults. They will die from this bacterial infection in their lungs, which pretty much, again, treated with antibiotics, but then bacteria will develop resistance and no antibiotics will help for these children to clear up their lungs from bacterial infections. So this is a big problem. So lots of research has been done, but there's been some progress, but very slow progress. Okay, so there is a ATPases. Now, let me see. I would like to, maybe you can read these, you can read this on by yourself, especially because it's studied in great detail in metabolism class. So we're not going to cover much of these, but there is one page of this in the textbook. It's page 416. So please read about these ATPases. There is some general description of how they work, but we're going to skip this and talk about aquaporins. So aquaporins are basically channels which allow for water to pass through cell membrane, right? Again, water is polar, doesn't easily cross the cell membrane. And so here we have a number of alpha helices. You can see there is an internal channel where the molecules of water are lining up. So there's lots of molecules of water here. Hold on a second. Lots of molecules of water here. And so the way this works, so this channel is highly specific for water, and that's primarily due to the makeup of the amino acids at this channel area of the transporter. So here, for example, this histidine 180, right? It kind of makes the channel area just wide enough for the molecules of water to squeeze through. Now, for example, if you have urea, right, which is a slightly bigger molecule, also capable of lots of hydrogen bonding, and many of the properties of urea are similar to water, but urea will not slide through, right? So the size of the channel is important. Then here, there is a water dipole reorientation. So there are these two asparagines, 76 and 192. And so what they do, they basically, they reorient the molecules of water in such a way that, so normally, obviously because of hydrogen bonding, the molecules of water will prefer to orient themselves like this. right? And so what this can result in, unfortunately, in a phenomenon known as proton hopping. Proton hopping, when basically oxygens will stay in one place, but instead of moving water, this system will move protons from one side to the other. And so that is precluded due to reorientation of this. So in other words, this will be broken and this molecule of water will undergo some kind of rotation to something like this. Right? Reorientation, so that there's so that the oxygens of this molecule of water do not interact with hydrogens of that molecule of water. So basically breaking up the hydrogen bonding between the molecules of water. Now this arginine 192 is positively charged. So this would repel, for example, hydronium ion, right? So this is responsible for to stop hydronium ion to migrate. And so it's very well designed. And in fact, it works so fast that allows for the rate of this is 10 to the 9th per second. So 10 to the 9 molecules of water per second. And remember, even for the best enzymes that process substrates, it's 10 to the 7th per second. So here we're a couple orders of magnitude faster than some of the fastest enzymes. So these aquaporins have been really designed by nature to allow for water to come in and out of the cells very quickly. All right. And the last thing is I wanted to briefly tell you are ion channels. So ion channels, for example, this one is a potassium channel. So it has also alpha helices as transmembrane domains. If we look from this view, you can see there's a potassium sitting right there in the middle of these transmembrane domains. And so the way this works is, again, potassium is surrounded by water hydration shell, right? It comes into this cavity where it shakes off the hydration shell, and water's actually replaced by the carbonyl groups. You see these red ones, red oxygens, are the carbonyl groups from the polypeptide or from these alpha helical segments on the ion channel, right? And so that allows for replacement of water by the carbonyl groups allows for the easy transport of potassium. And also these carbonyl groups, they're arranged in such a way that they can control the size. For example, sodium is too small, right? Sodium is too small and it will not form very strong interactions with the carbonyl. And if this sodium cannot form interactions with the carbonyl, it cannot get rid of its hydration shell, right? So sodium will not pass through easily. There's a huge selectivity between potassium and sodium. And once it gets out, then it basically regains its hydration shell. All right, so now these tables are just a compilation of different transport systems and their location in the textbook. You can see actually most of them are actually going to be studied in metabolism class, right? And so, so what did we learn? We learned that the lipids can form micelles, bilayers, liposomes. membranes, lots of different types of lipids, lots of different types of proteins. Properties depend on lipid composition, and they vary from organism to organism, tissue to tissue, organelle to organelle. Membrane proteins, 3 major classes, right? And many of them play a role in transport of solutes across the membrane. Active transport of solutes across membrane requires ATP, but can be accomplished in many different ways, in co-transport, symport, and so on and so forth. All right. Do you guys have any questions about chapter 11? Was everything clear today? Yeah? All right.
Yes.
I see some thumbs up. Sounds good. So the homework is open, as you know, right? Until Monday. And on Monday, we will start chapter 12. So we have 2 more lectures left. Monday, the 23rd, and Monday, the 30th, right? After Thanksgiving. And then we're done. All right, sounds good, guys. Let me know if you have any questions and write something in the discussion forum. Have a good rest of the week and good weekend.